Hello everyone, we're going to be covering chapter 21 today. We're going to be looking at the immune system. So we're going to be looking at innate and adaptive body defenses. So there are two main types of immunity that we will be discussing in today's chapter. They are innate immunity, also called non-specific resistance, and adaptive immunity. So innate immunity is called non-specific resistance because it covers a wide range of pathogens and it doesn't require learning or your immune system learning about the pathogen before it can combat it. This type of immunity is present at birth and it's more of really a general protection. Adaptive immunity, on the other hand, this is the type of immunity that we're actually kind of talking about when we typically think of the immune system. So this type of immunity involves an adaptation of lymphocytes, which remember are a type of white blood cell, to combat very particular pathogens. So this is much more of a specific immunity. Now, these different types of immunity are carried out by the lymphatic system. So let's first discuss the innate immunity. So this non-specific immunity includes the external physical as well as chemical barriers provided by the skin and mucous membranes. It also includes various internal defenses, such as antimicrobial substances, natural killer cells, phagocytes, inflammation, and fever. So these are going to be our internal defenses. So again, these are things like antimicrobial substances, natural killer cells, phagocytes, inflammation, and even fever. So really the innate immunity overall is going to be the type of defense that we are born with. So the skin and mucous membranes of the body are the first line of defense against pathogens. These structures provide both physical and chemical barriers that discourage pathogens and foreign substances from penetrating the body and causing disease. The epidermis of the skin provides the most formidable physical layer or physical barrier to the entrance of microbes. So that would be the skin, would be our first main mechanical defense. In addition, this periodic shedding of the epidermal cells helps to remove microbes at the skin surface. The epithelial layer of mucous membranes that lines body cavities and secretes mucus. This helps to lubricate and moisten the cavity surface. But also things like hair, cilia, saliva, and tears. These are all other mechanical defenses that we're born with. Some of our chemical defenses that we're born with include sebum, which is created by the sebaceous glands of the skin, lysozyme, which is an enzyme contained in tears, as well as gastric juice in the stomach, all work as a chemical defense. The second line of defense, if pathogens get past that first line of mechanical and chemical barriers, is this internal defense. So this second line of defense includes internal antimicrobial proteins, phagocytes, natural killer cells, even inflammation and fever are included in our innate internal defense. Now, when we're looking at antimicrobial substances that discourage microbial growth, there's going to be four main types. So our first one are interferons. So lymphocytes, macrophages, and fibroblasts that are infected with viruses produce these proteins that are called interferons. Often they're abbreviated IFs. Now these are proteins made by these infected cells that interfere with viral replications. Interfere. Our second main type of antimicrobial substances, is complement. So we have this group of normally inactive proteins in blood plasma that make up this complement system. And these are going to be proteins that enhance certain immune reactions. Our third antimicrobial substance is iron binding proteins. So these are going to be proteins that inhibit the growth of certain bacteria by reducing the amount of available iron. And these certain bacteria require iron to grow. We will talk about each of these individually too, but just a quick overview yet. Well, we'll talk about complement for sure, but these are just an overview of the rest of them. Our last one are called antimicrobial proteins. So these are also abbreviated AMPs. These are really short peptides that have a broad spectrum of antimicrobial activity. When microbes penetrate the skin and mucous membranes or bypass the antimicrobial substances in the blood, the next nonspecific defense consists of natural killer cells, or NK cells, and phagocytes. So about 5 to 10% of lymphocytes in the blood are natural killer cells. they will lack the membrane molecules that identify B and T cells, but they have the ability to kill a wide variety of infected body cells and even certain tumor cells. Natural killer cells will attack any body cells that display abnormal or unusual plasma membrane proteins. Proteins. Okay, now phagocytes, on the other hand, these are going to be specialized cells that perform phagocytosis. So phagocytosis is that ingestion of microbes or other particles like cellular debris. I'll put other particles. Now, there are two major types of phagocytes. These are neutrophils and monocytes. Now, macrophages will actually develop from monocytes. And when an infection occurs, neutrophils and monocytes will migrate to the infected area. In addition to being an innate defense mechanism, phagocytosis really plays a vital role in adaptive immunity that we will talk about later as well. Now on this picture on the right, we have the phases of phagocytosis. So this is a phagocyte, either a neutrophil or a mature macrophage. So monocytes are actually going to be the immature cell type, and monocytes are actually not capable of phagocytosis, but it's only when they differentiate or develop further into macrophages when they're able to go through phagocytosis or conduct phagocytosis once they are macrophages. So here is our first phase of phagocytosis, which is chemotaxis. So this is a chemically stimulated movement of phagocytes to a site of damage. Chemicals that attract phagocytes may come from invading microbes, white blood cells, damaged tissue cells, or activated complement proteins. And so these little chemicals that we can see here actually kind of attract the phagocyte to this area. So that's going to be the actual definition of chemotaxis. Chemically stimulated movement of a phagocyte to a site of damage. So usually to a site of damage or also to a microbe or other foreign particle. Our second step of phagocytosis is adherence. So this is the part where the phagocyte attaches to the microbe or other foreign material. This step is where the binding of complement proteins to the invading pathogens enhances adherence. So we can add this here. So complement protein enhances this step. We'll learn more about complement later, but these are specific proteins that can bind to the microbes and enhance adherence. Our step 3 is ingestion. So this is where the plasma membrane of the phagocyte will extend little projections, which are called pseudopods, that engulf the microbes in a process called ingestion. When those pseudopods go around the microbe and they meet at the other end, they fuse together, surrounding the microorganism with a sac called a phagosome. Step 4 of phagocytosis is digestion. So the phagosome will enter the cytoplasm and merge with lysosomes to form a single larger structure called a phagolysosome. You can kind of see this structure here. So that's a phagolysosome. So the lysosome contributes lysozyme, which breaks down the microbial cell walls, and then there's going to be a lot of other digestive enzymes in the lysosome as well that break down carbohydrates, proteins, lipids, and nucleic acids. So here we have that digested microbe in the phagolysosome. Then our final step of phagocytosis is the killing phase. So the chemical onslaught of lysozyme, digestive enzymes, and even some oxidants within the phagolysosome will very quickly kill many types of microbes. Any materials that cannot be degraded further remain in these structures called residual bodies. So these have these indigestible material. Now here we have inflammation. And inflammation is a non-specific defensive response of the body to tissue damage. The four characteristic signs and symptoms of inflammation are redness, pain, heat, and swelling. Because inflammation is one of the body's non-specific defense mechanisms, the response of a tissue to a cut is similar to the response to damage caused by burns or radiation or even bacterial or viral infection. In each case, the inflammation response has 3 basic stages. So these are going to be the stages of inflammation here. So our first stage of inflammation is going to be vasodilation and increased permeability of the blood vessels. I'm just going to do up arrow, increased permeability of the blood vessels. So we can see this first step here on our picture on the right. So here we have a cut tissue injury in the skin. And here we have our first stage, vasodilation and that increased permeability of the blood vessels. Our stage number 2 is emigration of phagocytes from the blood into the interstitial fluid. So you can see that step right here, that emigration of these phagocytes. So emigration, remember, is just movement or the movement of these cells out of the blood vessel. And then our final stage of inflammation is tissue repair. So just a little bit of discussion about these stages. So 2 immediate changes occur in blood vessels in a region of tissue injury. So these are going to be that vasodilation and increased permeability. So vasodilation means that there's an increase in diameter of the arterioles. So here you can kind of see diameter this big, but here near the tissue injury we have a diameter maybe about that big. So much larger diameter of those arterioles near the injury. And increased permeability of the blood vessels means that substances that are normally retained in blood are permitted to pass from the blood vessels. And vasodilation really just allows more blood to flow through the damaged area. So both of these are very important in our first stage of inflammation. So this increased permeability, it's really going to allow for defensive proteins like antibodies and clotting factors to enter the injured area from blood. So we're going to have like outflow here of not only these phagocytes, but also things like antibodies and clotting factors to leave the blood here. But this increased blood flow will also help to remove microbial toxins and dead cells from the area too. Now pain, this is a very prime symptom of inflammation. It really results from the injury to neurons as well as toxic chemicals that are released from the microbes in the area. Within an hour after the inflammation process starts, the phagocytes will appear on the scene. So as large amounts of blood accumulate, the neutrophils will begin to stick to the inner surface of the endothelium of the blood vessels. And then those neutrophils will begin to squeeze through between those endothelial cells in order to reach the damaged area. So this process we know is emigration, and this depends on large part depends on chemotaxis. So there's chemicals in the area that are attracting those phagocytes to the area. Although neutrophils really predominate the early stages of infection, they die off really quickly, and eventually the macrophages will also die. Within a few days, there's going to be a packet of these dead phagocytes and damaged tissue that will form. And the collection of dead cells and damaged tissue, fluid, this is all called pus. This table here summarizes that first line of defense, the skin and mucous membranes. So we're going to get all of those physical factors we kind of talked about, the skin, mucus, hair, saliva, and it will give an outline of the functions of each of them as well. We're also going to get those chemical factors, sebum and lysozyme, as well as the gastric juice and vaginal secretions are all on this list as well. And then this table summarizes the second line of defense, those internal defenses. So keep in mind that this second line of defense is still innate defenses that you're born with. So we're not talking about adaptive immunity yet, we're still talking about the innate immunity. So here are those antimicrobial substances, natural killer cells and phagocytes, as well as the inflammation and fever. So again, these are all things that we've talked about, but nice to have a nice summarizing table here. The ability of the body to defend itself against specific invading agents such as bacteria, toxins, viruses, and foreign tissues is called the adaptive immunity, also known as specific immunity. Substances that are recognized as foreign and provoke immune responses are called antigens. Specifically, it means antibody generators. So antigen. Now there are two properties that distinguish adaptive immunity from innate immunity. These are the specificity for particular foreign molecules or antigens, which involves distinguishing self from non-self. So that means that your adaptive immunity will be able to determine which cells are your cells and which cells coming in are either from something entirely different or another person, anything non-self. Our second property that distinguishes adaptive immunity from innate immunity is going to be the memory for most previously encountered antigens, so that a second encounter prompts an even more rapid and vigorous response. So that memory that your cells have when it encounters an antigen really helps that when you have a second encounter with those same antigens, your adaptive immunity will be much quicker to respond and the response will be much more heightened. Now, adaptive immunity has two different types. There are cell-mediated adaptive immunity and antibody-mediated adaptive immunity. Adaptive immunity involves lymphocytes called B cells and T cells. Now, both of these cells develop in the primary lymphatic organs, red bone marrow and the thymus. And they develop from pluripotent stem cells that originate in red bone marrow. T cells will mature in the thymus, whereas B cells complete their development in red bone marrow. Now, before T cells leave the thymus or B cells leave the red bone marrow, they develop immunocompetence. So remember that immunocompetence is the ability to carry out adaptive immune responses. So this means that B cells and T cells begin to make several distinct proteins that are inserted into their plasma membranes. Now, some of these proteins function as antigen receptors. So these are molecules capable of recognizing specific antigens. We'll talk about a little bit more about these antigen receptors in a little bit. Now, there are two major types of mature T cells that exit the thymus. These are helper T cells and cytotoxic T cells. Helper T cells are also known as CD4 T cells, which means that in addition to their regular antigen receptors, their plasma membrane also includes a protein which is called CD4. So this is kind of how we recognize helper T cells, by this CD4 protein. Cytotoxic T cells, these are also referred to as CD8 proteins, or CD8 cells, sorry, and this is because their plasma membrane has these proteins called CD8 proteins. This is also in addition to their antigen receptors. These two types of T cells have very different functions. We'll discuss these a little bit more a little bit later. Now, we already know that there's two types of adaptive immunity. These are cell-mediated immunity and antibody-mediated immunity. Now, both types of adaptive immunity are triggered by antigens. So both types of immunity are triggered by antigens. In cell-mediated immunity, this left column here, this is where cytotoxic T cells directly attack invading antigens. So this is kind of our end product. These activated cytotoxic T cells will directly attack invading antigens. On the other hand, on our right column where we have our antibody-mediated immunity, this is where B cells up here, they're going to transform into plasma cells, which will synthesize and secrete specific proteins that we know as antibodies. I guess they're labeled here too. So this is where the antibodies will bind to antigens. So let's just briefly go through the top here. We can also see these antigen receptors labeled here at the top. Now, so we know that cytotoxic T cells become activated and will directly attack invading microbes. What about these helper T cells? So helper T cells will aid the immune responses both in cell-mediated and in antibody-mediated immunity. So helper T cells will become activated – and they will help to activate cytotoxic T cells, which will where we get that activation of the cytotoxic T cell. And they will also help to activate the B cells in antibody-mediated immunity. So helper T cells, just like their name implies, are going to help other cells to become activated. Now we also see these other cells here, memory helper cells, memory cytotoxic T cells, and memory B cells. So what are those all about? So memory cells do not actively participate in the initial immune response to the antigen. However, if the same antigen enters the body again in the future, the thousands of memory cells of a lymphocyte clone are available to initiate a really much faster reaction than occurred during the first invasion. So if we have memory B cells, they already have come into contact with an antigen prior, then they'd be able to become activated very much quicker than the first time we came into contact with that antigen. Antigens have two very important characteristics. These are immunogenicity and reactivity. So immunogenicity is the ability to provoke an immune response by stimulating the production of antibodies or the proliferation of specific T cells or both. So stimulating that production of specific antibodies. So antibodies, I'll just abbreviate ABs. So the proliferation, again, of specific T cells. Or both. Now, reactivity is the ability of the antigen to react specifically with those antibodies or cells it provoked. So antigen, I'm just going to abbreviate AG. A huge variety of antigens in the environment provides many opportunities for provoking immune responses. Typically, just certain small parts of a large antigen molecule actually act as the triggers for immune responses. So these small parts that act as the triggers are called epitopes. These are also called antigenic determinants. Most antigens have very many epitopes, each of which induces production of a very specific antibody or activates a specific T cell. So here we can see a variety of very different epitopes on this antigen. Now, for an immune response to occur, B cells and T cells have to recognize that a foreign antigen is even present. So B cells can recognize and bind to antigens in lymph, interstitial fluid, or blood plasma. T cells will only recognize fragments of antigenic proteins that are processed and then presented to the T cells in a very specific way. In antigen processing, the antigenic proteins are broken down into peptide fragments that will then associate with specific molecules that are called MHC molecules. MHC stands for Major Histocompatibility Complex. So it's going to be that MHC. So MHC molecules are located in the plasma membrane of body cells. Essentially, they're going to be known as self-antigens. So unless you have an identical twin, your MHC antigens are unique to you, and they allow your other cells to differentiate between self and non-self. So this is an important recognition in immune responses, as you don't want your white blood cells attacking your own body cells. So they have to be able to differentiate which are self cells, which are your own cells, and which are non-self cells. So back to the antigen processing. Once the peptide fragment is broken down and associated with an MHC molecule, it makes this antigen MHC complex, and this complex is inserted into the plasma membrane of the cell. Now, the insertion of the complex into the plasma membrane, this is called antigen presentation. Foreign antigens that are present in fluids outside body cells are called exogenous antigens. And a special class of cells, which are called antigen-presenting cells, or APCs, will process and present exogenous antigens. APCs include dendritic cells, macrophages, and B cells. So these are all just different types of antigen-presenting cells. So the steps in processing and presenting of an exogenous antigen by an antigen-presenting cell occurs like this in this picture here. So at step 1, we have the ingestion of the antigen. So this is going to occur by phagocytosis or endocytosis. Step 2, we have the digestion of the antigen into peptide fragments. So when the antigen is ingested, it gets put into a phagosome if it was phagocytized, or an endosome if it was taken in by endocytosis. And then the antigen is digested into small fragments. So within the phagosome or the endosome, there's going to be protein digestion enzymes that will split large antigens into short peptide fragments. Step 3 is down here at the bottom of this cell, and this is the synthesis of MHC2 molecules. So this is going to happen at the same time that the antigen is being engulfed by the cell. This is where the APC is going to synthesize MHC2 molecules, and it's going to make them in the endoplasmic reticulum. So this is an important note that exogenous antigens, these are presented on MHC MHC2 molecules. This will be important when we look at the next slide when we're looking at endogenous antigens. They're presented differently, so remembering that exogenous antigens will combine with MHC2 molecules. So once those MHC2 molecules are created, we package them. So in step 4 here, MHC2 molecules are going to be packaged into vesicles. Then those vesicles containing antigen peptide fragments and the MHC2 molecules will merge together and fuse. So they're going to be in the same vesicle now. So that's step 5, fusion of the vesicles. Then we get to step 6 here. This is where we get binding of the peptide fragments to the MHC molecules. So after fusion of these two types of vesicles, the antigen peptide fragments will bind to the MHC2 molecules. Then our final step is the insertion of the antigen MHC2 complex into the plasma membrane. So the combined vesicle that contains the antigen MHC2 complex will undergo exocytosis. And as a result, they're going to be inserted into the plasma membrane. So in this way, they're kind of presenting the antigen peptide fragments on the surface of their plasma membrane. So we can see that presentation right here. So the presentation of exogenous antigens together with the MHC2 molecules by antigen presenting cells will inform T cells that intruders are present in the body and that combative action should begin. So they're going to present this to T cells. Now, when a peptide fragment comes from a self protein, T cells will ignore the antigen MHC complex. So if it's from a self protein peptide fragment, then the T cells will ignore it. But if it's from anything else other than a self peptide, then the T cells know that we have to kind of set up a combative action, essentially. And to start fighting off this exogenous antigen because it's presented with an MHC2 molecule. Foreign antigens that are present inside body cells are called endogenous antigens. Now, such antigens may be viral proteins produced after a virus infects the cells and takes over the cell's metabolic machinery. So in this picture here, we have the steps in the processing and presenting of an endogenous antigen by infected body cells. So this is any infected body cell can present an endogenous antigen. So our first step here is going to be digestion of the antigen into these peptide fragments. So protein digesting enzymes will split the endogenous antigen into short peptide fragments. So that's going to be our first step. Our step number 2 is down here at the bottom, which is the synthesis of the MHC1 molecule. So this is at the same time as we're digesting the peptide or digesting the antigen into peptide fragments, the infected cell will synthesize MHC1 molecules in the endoplasmic reticulum. So again, something important to remember that endogenous antigens are going to be presented on MHC1 molecules. Now our step 3 is the binding of peptide fragments to MHC1 molecules. So the antigen peptide fragments will actually enter into the endoplasmic reticulum and then bind to those MHC1 molecules. Step 4 is the packing of an antigen MHC1 molecule. So from the ER, the antigen MHC1 molecules are packaged into vesicles. And then our final step here is step 5, where we get insertion of the antigen MHC1 complex into the plasma membrane. So the vesicles that contain antigen MHC1 complexes will undergo exocytosis, and this will insert them into the plasma membrane. And so this is going to be the way that they present these endogenous antigens onto the plasma membrane. So most cells of the body can process and present endogenous antigens. So the display of an endogenous antigen that's bound to an MHC1 molecule will signal that the cell has been infected and it needs help. Cytokines, these are small protein hormones that stimulate or inhibit many normal cell functions such as cell growth or differentiation. Lymphocytes and antigen-presenting cells will secrete cytokines. So do fibroblasts, endothelial cells, monocytes, hepatocytes, even kidney cells will secrete cytokines. So lots of different cells will be able to secrete these cytokines. Some cytokines will stimulate the proliferation of progenitor blood cells in red bone marrow. Some will regulate the activities of cells involved in innate defenses or adaptive immune responses. So this table here describes 8 types of cytokines and their functions. So on the left we have the cytokines, on the right the origins and functions. We'll talk about some of these in the future, so you can probably come back to this table a little while later, but we'll definitely talk about interleukin-2. We'll talk about the tumor necrosis factor and interferons. So definitely a good table, maybe to just put a pin in this, and we can come back to this later when we kind of talk about these specific cytokines as they come up in our chapter. A cell-mediated immune response begins with the activation of a small number of T cells by a specific antigen. Now, once a T cell has been activated, it undergoes clonal selection. So this is after it's been activated. So activated T cell will then go through clonal selection. Now, clonal selection is a process by which a lymphocyte proliferates and differentiates in response to a specific antigen. So it's going to proliferate, which means it divides several times. And it will also differentiate, which means it forms more highly specialized cells. Now this is in response to a specific antigen. So they're going to differentiate into a cell that recognizes a specific antigen, and then it will divide several times so that we get these clones that all recognize a specific antigen. Now the effector cells of a T cell clone will carry out immune responses that ultimately result in the elimination of the intruder. So we're going to have this antigen is recognized and bound, which activates that T cell. And then a number of T cells will proliferate and differentiate into clone effector cells. Again, those are going to be T cells that were cloned to recognize this specific antigen. And then in this way, the antigen will be eliminated. At any given time, most T cells are inactive. Now remember that antigen receptors on the surface of T cells, which are called T cell receptors, will recognize and bind to specific foreign antigen fragments that are presented in antigen MHC complexes. So T cell receptors, these are also called TCRs. These will recognize and bind to these antigen fragments. So these are ones that are presented by MHC molecules. Now there are millions of different T cells. Each of them has their own unique T cell receptors that can recognize a specific antigen MHC complex. Now a T cell becomes activated only if it binds to the foreign antigen and at the same time receives a second signal, which is known as co-stimulation. So T cell activation, this is going to require binding to the antigen, plus receiving a second signal. So these two together would be co-stimulation. Now, of the 20, well, there's more than 20 known co-stimulators. Some of them are cytokines, one of them being interleukin-2. This acts as a co-stimulator, or it acts as that second signal to help activate T cells. So interleukin-2, one of those cytokines that we looked at before, is a very common co-stimulator. Now remember, most T cells that display this CD4 protein develop into helper T cells. So remember, these are also known as CD4 T cells. Now, inactive helper T cells, these are also known as just like resting helper T cells, these will recognize exogenous antigens that are associated with MHC2 molecules. And this has to be on the surface of an antigen presenting cell. So remember, antigen presenting cells, APCs, will present exogenous antigens. on MHC2 molecules. Now, with the aid of this CD4 protein, the helper T cell and the APC will interact with each other. This is called antigen recognition. So this is just this interaction with the antigen presenting cell and the inactive helper T cell, where the T cell is going to recognize that MHC2 antigen complex. Then we have co-stimulation occur, and the helper T cell becomes activated. Now, once activated, the helper T cell will undergo clonal selection. The result is a formation of a clone of helper T cells that consist of active helper T cells and memory helper T cells. So we kind of get both of them. Within like hours of co-stimulation, the active helper T cells will start secreting a variety of cytokines. One very important cytokine produced by helper T cells is going to be that interleukin-2, which is really needed for virtually all immune responses and is the prime trigger of T cell proliferation. Now, most T cells that display the CD8 protein develop into cytotoxic T cells, also known as CD8 T cells. Now, cytotoxic T cells, with their T cell receptor, will recognize foreign antigens combined with major histocompatibility complex class 1, also known as MHC1 molecules. And these molecules will present antigens on a couple of different cells. So body cells that are infected by microbes, some tumor cells will also present antigens on an MHC1 molecule. And also cells of a tissue transplant. So essentially, cells that have come from another person. These are going to be non-self cells. Now, recognition requires the TCR and the CD8 protein to maintain the coupling with the MHC1 molecule. This is where we get antigen recognition. Pretty much this little circle here is going to be all that's required for antigen recognition. The MHC1 molecule presents the antigen, and the TCR recognizes that MHC1 antigen complex, and then the CD8 protein is also involved in that antigen recognition, and then co-stimulation will occur. Now, in order to become activated, cytotoxic T cells require co-stimulation by interleukin-2 or other cytokines produced by activated helper T cells that have already become bound to copies of the same antigen. So these helper T cells have already bound to copies of this purple antigen here. Now remember that these helper T cells are activated by antigens associated with MHC2 molecules. Now, once activated, so here we have this activated cytotoxic T cell, is going to undergo that clonal selection. And the result is the formation of those cytotoxic T cell clones. That consists of these active cytotoxic T cells and also memory cytotoxic T cells. Now, active cytotoxic T cells will attack other body cells that have been infected with that antigen that they became activated by. Memory cytotoxic T cells do not attack infected body cells, but instead they can quickly proliferate and differentiate into more active cytotoxic T cells and more memory cytotoxic T cells if the same antigen enters the body again. So these memory cytotoxic T cells, these will quickly proliferate and differentiate and produce more of these active cytotoxic T cells and memory cytotoxic T cells if the same antigen enters the body again. It's the cytotoxic T cells that are the cells that go and do battle in your body and combat the invaders. They leave the secondary lymphatic organs and migrate out to seek and destroy infected target cells. Now, cytotoxic T cells will kill infected cells in much the same way that the natural killer cells do. The major difference is that cytotoxic T cells have receptors specific for a certain microbe, and so they're only going to kill target cells that are infected with that one particular microbe. Natural killer cells, on the other hand, will destroy a wide variety of microbe-infected body cells. So cytotoxic T cells have two principal mechanisms for killing infected target cells. Our first one is here on the left. This is where the activated cytotoxic T cells, using receptors on their surfaces, will recognize and bind to infected target cells that have these microbial antigens displayed on their surface. So we can see this MHC molecule with the antigen being presented on the infected body cell surface. And that activated cytotoxic T cell will recognize that and bind to the infected body cell. Once that recognition occurs, the T cell will then release granzymes. These are protein digesting enzymes that trigger apoptosis. So these granzymes will come into contact with the infected body cell, and that infected body cell will undergo apoptosis. Now, once the infected cell is destroyed, the released microbes are going to be killed by phagocytosis of phagocytes in the area. Alternatively, on the right here, the cytotoxic T cell will bind to infected body cells and release two proteins from their granules. These are perforin and granulysin. Perforin will insert into the plasma membrane of the target cells and creates channels in the membrane. Other granules and cytotoxic T cells will release granulysin, which will enter through those channels created by the perforin. So the granulysin is going to go into the infected body cell through those perforin channels, and it destroys the cell by creating holes in the plasma membrane. So then we get this cytolysis, essentially cell explosion kind of thing, or cell bursting, you could say. Essentially, both of these methods will destroy the cell, causing cytolysis. Now, the body contains not only millions of different T cells, but also millions of different B cells. Each is capable of responding to a specific antigen. Now, cytotoxic T cells will leave lymphatic tissues to seek out and destroy a foreign antigen, but B cells, they stay put. In the presence of a foreign antigen, a specific B cell in a lymph node, the spleen or mucosa-associated lymphatic tissue, will become activated. It will undergo clonal selection. So these helper T cells will activate the B cell, go through that clonal selection of proliferation and differentiation, and they will form clone plasma cells. Plasma cells are kind of the activated form of B cells. So if we have a B cell, and it becomes activated, it becomes a plasma cell. So plasma cells are going to be the effector cells of a B cell clone, and they secrete specific antibodies. These antibodies will circulate in the lymph and the blood to reach the site of invasion. So B cells will essentially stay in their areas. They'll stay in the lymph nodes, in the spleen, or in those MALT, that mucosa-associated lymphatic tissue. So once they become activated, they turn into plasma cells. Those plasma cells will secrete very specific antibodies, specific to the antigen that's recognized, so that those antibodies will flow through the lymph and the blood to eliminate the antigen. During activation of a B cell, an antigen will bind to B cell receptors. These are known as BCRs as well. Now, these are integral transmembrane proteins, and chemically they're going to be very similar to the antibodies that are later secreted by plasma cells. You see, they kind of do look the same on this picture, save for the color, but B cell receptors are not the same as antibodies. The antigen is taken into the B cell, broken down into peptide fragments, and then it is combined with MHC2 self-antigens, and then it's moved to the B cell plasma membrane. So we can see one right here. Now, helper T cells will recognize the antigen MHC2 complex, and deliver the co-stimulation needed for B cell proliferation and differentiation. So that's where we get that clonal selection, because that helper cell, or the helper T cell, gives co-stimulation, allowing for clonal selection of these activated B cells. So once these B cells are activated by co-stimulation, they go through clonal selection, just like T cells, to produce plasma cells, which secrete antibodies, and memory B cells. Now, memory B cells do not secrete antibodies, but instead, just like memory T cells, these memory B cells can quickly proliferate and differentiate into more plasma cells and more memory B cells should that same antigen reappear at a future time. Antibodies belong to a group of glycoproteins, which are called globulins. And for this reason, these are also called immunoglobulins. Often abbreviated IG. Is for a singular immunoglobulin. Now, most antibodies contain 4 peptide chains. 2 of these chains are identical to each other. These are called heavy chains. So we have a blue heavy chain and a yellow heavy chain on this particular antibody. And the other 2 peptide chains, or polypeptide chains, which are also identical to each other, are called light chains. So we have a red light chain on either side of this immunoglobulin. Now the top of the Y of the antibody, this is the antigen binding site. This is also known as the variable region. And this part of the antibody is going to be different for each kind of antibody because it is specific to the antigen. This is the part of the antibody that recognizes and attaches specifically to a particular antigen. Now these variable regions are abbreviated with V's since they are variable. Now the remainder of each heavy and light chain, the bottom part of the Y, these are going to be called the constant region. And this constant region, abbreviated with C's here, you can see all these C's. These are going to be nearly the same in all antibodies of the same class. And this part of the antibody is responsible for the type of antigen-antibody reaction that occurs. So essentially, the constant region is responsible for how we're going to combat a specific antigen. So there are actually 5 classes of immunoglobulins, or 5 classes of antibodies. These are IgG, IgA, IgM, IgD, and IgE. Now, the actions of the 5 classes of immunoglobulins will differ somewhat, but all of them act to disable antigens in some way. Now, actions of antibodies will include things like neutralizing the antigen. I can write this in the bottom here, but these are just specific things that all of these can do, or would be one of their functions. So neutralize the antigen could be one action that they can do. So the reaction of the antibody with the antigen will block or neutralize some bacterial toxins and can even prevent the attachment of some viruses to body cells. So in this way, they would neutralize the antigen. Another action could include immobilizing the bacteria. So if antibodies form against antigens on the cilia or flagella of motile bacteria, it may cause the bacteria to lose their motility. Hence, it would immobilize them. They may also agglutinate the antigen. Or clump the antigen together. So because antibodies actually have two sites for binding an antigen, or sometimes they have way more sites for binding antigen, the antibody reaction may cross-link the pathogens to one another. So say we have one antigen here, it's binded to that IgM, but then there's another IgM is bound to it here, like this. And then we have another antigen here, which is bonded to this part of the IgM. And then this one is also bound to another IgM, for example. So we're going to be clumping all of these antigens together because one antigen can bind with several antibodies. Also, when antigens become agglutinated because they're bonded to many antibodies, they also become phagocytized a lot more easily. So it's a really easy way to clean up antigens when they're bonded to antibodies. Another action of antibodies includes activating complement. So antigen-antibody complexes can initiate the classical pathway of the complement pathway that we'll actually talk about in the next slide. So this is another thing that antibodies can do. And the last one is enhancing phagocytosis. So the stem region, kind of this part down here, the stem region of an antibody acts as a flag. So this part is going to act as this massive flag here. And it acts as a flag that attracts phagocytes once antigens have bound to the antibody's variable region. So the stem region of the antibody will actually flag down phagocytes to engulf it once it has bound to an antibody. So this table here is really cool. It shows those 5 classes of antibodies and their specific characteristics and functions. So we see here that IgG is going to be the most abundant of all the antibodies. About 80% of all the antibodies in blood are going to be the IgG class. They protect against bacteria and viruses by enhancing phagocytosis. So that's this role here. They also work to neutralize toxins. So they're going to neutralize the antigen. And also work by triggering the complement system, activating complement. So this is going to be a very important antibody class. that does several of the actions that antibodies can do. One other very important thing about IgG is that it's the only class of antibody that can cross the placenta from the mother to the fetus. So in this way, we give considerable immune protection to newborns. So whatever the mother becomes immune to because of these antibodies, it will cross the placenta and also add that immunity to the fetus. IgA, found mostly in sweat and tears, saliva, mucus, and breast milk, as well as gastrointestinal secretions. Provides this localized protection of mucous membranes against bacteria and viruses. IgM is also present in kind of the second largest amount, or I guess third, because IgA is about 10 to 15 here. IgM, yeah, about 5 to 10% of the antibodies in blood. They're also going to activate complement, cause agglutination, as well as lysis of microbes. These are going to be anti-A and anti-B antibodies of the ABO blood group. Moving on to IgD, these are really on B cells. They're mostly involved in the activation of B cells. Very small amounts of antibodies of these IgD class, only about 0.2% here. And then IgE is also found in very small amounts, but this one has to do a lot with allergic reactions. So very, very important antibody for immunoreactions, or allergic reactions rather. Also provides some protection against parasitic worms. On this slide, we're going to go over the complement system. So the complement system, this is a defensive system that's made up of over 30 proteins that are produced by the liver. And these proteins are found circulating in blood plasma all the time and within tissues throughout the body. Now collectively, the complement proteins will destroy microbes by causing phagocytosis, cytolysis, and inflammation. Those are going to be the three kind of outcomes of the complement system. So phagocytosis, cytolysis. Remember, cytolysis is going to be that cell bursting, and then inflammation. The complement system also acts to prevent excessive damage to body tissues. Okay, so most complement proteins are going to be designated with an uppercase letter C, and they're labeled from C1 to C9. And they're named again in the order that they were discovered, not in the order that they are activated in the complement system. So that the numbering can kind of get a bit confusing, but just remember that they're numbered in the order that they were discovered. Now the C1 to C9 complement proteins are inactive. And really they only become activated when they become split by enzymes, and they're split into these active fragments. So once they become activated, they're indicated by lowercase letters A and B. So these are going to be inactive if they're just the uppercase, but if we have like C1A to, wow, I was going to say C9, yeah. But C1A, for example, is an activated complement protein. Now, complement proteins work in a cascade. So one reaction is going to trigger another reaction and so on and so forth. With each succeeding reaction, more and more product is formed so that the net effect is amplified many more times. So essentially, one complement protein will influence or activate many other complement proteins, and those are each going to activate many more, and so we get this cascading amplification effect. Now there are a couple different ways that complement activation may occur, but essentially all of them end with the activation of C3. So once C3 is activated, it begins this cascade of reactions that brings about either phagocytosis, cytolysis, or inflammation. So once C3 is activated, a couple of things happen. First off, the inactivated C3 is going to split up into an activated C3A and an activated C3B. C3b will bind to the surface of microbes, and receptors on phagocytes can attach to the C3b, which enhances phagocytosis. So this is by a coating of C3b. We also call this coating on microbes opsonization. So that's kind of where we get our first pathway or first outcome that leads to phagocytosis. Now C3b can also initiate a series of reactions to bring about cytolysis. So first, this activated C3b can split C5. So it'll split C5 into an active C5b and C5a. The C5b fragment will then bind to C6 and C7, which are here, which attach to the plasma membrane of, plasma membranes right here, of a microbial cell. So C5b will bind to C6 and C7 and attach to the plasma membrane of this invading microbe. Then C8 and C9 molecules will join the other complement proteins, and together they're going to form this cylinder-shaped membrane attack complex, which is going to insert into the plasma membrane. The membrane attack complex creates channels or pores in the plasma membrane that results in cytolysis. This is the bursting of the microbial cells, and this is due to the inflow of extracellular fluid through these channels or the outflow of the cytosol into the extracellular fluid. So that bursting microbe due to that inflow of extracellular fluid through the channel. So that's how it brings about cytolysis. Now our last outcome that complement can bring about is inflammation. So if we kind of go up a little bit more here, we have C3A and C5a will bind to mast cells and cause them to release histamine that increases blood vessel permeability during inflammation. So they're going to increase that blood vessel permeability. And C5a will also attract phagocytes to the site of inflammation by chemotaxis. So very important process there as well. A hallmark of immune responses is memory for specific antigens that have triggered immune responses in the past. Immunological memory, this is due to the presence of long-lasting antibodies and very long-lived lymphocytes that arise during clonal selection of antigen-stimulated B cells and T cells. Now, after initial contact with an antigen, no antibodies are present for a period of several days. So here we get that first exposure. We have a couple days where there's going to be no antibodies present. Then we get a slow rise in the antibody titer. This word titer, this is the amount of antibody in the blood serum. So first we get a rise in the IgM antibody and then IgG, followed by this gradual decline in the titers. So this is going to be our primary response. Now, memory cells may remain for decades. Every new encounter with the same antigen will result in rapid proliferation of memory cells. So after subsequent encounters, the antibody titer is far greater than during a primary response, and it mainly consists of IgG antibodies. So if here we get a second exposure, we're going to get a much more accelerated and more intense response, much higher antibody titer than our first exposure. So this more intense response is called a secondary response. Antibodies produced during a secondary response have an even higher affinity for the antigen than those that were produced during a primary response. So higher antigen affinity here too than in that primary response. So this table here summarizes the various ways to acquire adaptive immunity. Could be natural or artificially. So artificially, down here would be like by vaccines. And it could also be an active immunity or a passive immunity. So active would be by the transfer of antigens. Passive is by the transfer of antibodies. So naturally acquired passive immunity would be the transfer of antibodies from mother to fetus across a placenta. Whereas if we're artificially acquiring passive immunity, this is going to be an IV injection of antibodies. Naturally acquiring an active immunity is going to be exposure to a microbe. We get that antigen recognition by the B cells and T cells, co-stimulation, and then those activated T cells and B cells. Artificially acquired active immunity is going to be vaccination with antigens in it. So we're going to stimulate those cell-mediated and antibody-mediated immune responses. So different ways to kind of get the same or a very similar result. Now, to function properly, your T cells must have two traits. they must be able to recognize your own major histocompatibility complex proteins. This is a process known as self-recognition. So that's kind of this first trait that they have to have. So this is recognize your own major histocompatibility complex proteins. So this is self-recognition. Your T cells have to be able to do this. And our second trait that T cells have to do in order to function properly is they have to lack reactivity to peptide fragments from your own proteins. So this is known as self-tolerance. So lack reactivity to peptide fragments from your own proteins. So this is for T cells. Now, B cells, these will also display self-tolerance, and loss of self-tolerance leads to the development of autoimmune diseases. Okay, so here we have development of T cells on this slide. So pre-T cells in the thymus will develop the capability for self-recognition by a process called positive selection. So in this process, some T cells will express T cell receptors that interact with self-MHC proteins on the epithelial cells in the thymic cortex. Now, because of this interaction, the T cells can recognize the MHC part of the antigen MHC complex. So it's kind of they go through this process is if they recognize our self-MHC proteins. Now, the cells that do recognize our self-MHC proteins, these T cells survive because they can recognize self. Other immature T cells that kind of fail this interaction, they're going to undergo apoptosis. So this process here develops self-recognition. So that's what this process is developing that capability for self-recognition. Then we have to develop the self-tolerance. So this development occurs kind of by a weeding out process, which is called negative selection. In negative selection, the T cells will interact with dendritic cells located at the junction of the cortex and the medulla of the thymus. Now, in this process, we have are the T cells capable of binding to and recognizing self-peptides. So we don't want them to recognize self-peptides. We don't want them to kind of react to self-peptides. So if they don't react, then those cells survive. So this is a process to develop self-tolerance. If they do react, they are either going to be eliminated and deleted, or they may be inactivated, process called anergy. Now, once T cells have passed both, they come out of the thymus, they may still encounter an unfamiliar self-protein. Now, in these cases, they also may become anergic if there is no co-stimulation. So when we have an antigen without co-stimulation, like here, they'll still become anergic or inactivated. Deletion of self-reactive T cells may also occur after they leave the thymus and are mature, and this causes apoptosis. So these mature T cells that leave the thymus, it's only after they have antigen recognition with co-stimulation that they become an activated T cell, and then they go through clonal selection. B cells also develop tolerance through deletion and anergy. While B cells are developing in bone marrow, those cells exhibiting antigen receptors that recognize common self-antigens like MHC molecules or blood group antigens, they are going to be deleted. So if they recognize these self-antigens, they will go through apoptosis within the bone marrow. If they don't, this is a negative selection to develop tolerance, and they will continue on to the next step. Now, once B cells are released into the blood, after this point, anergy really appears to be the main mechanism for preventing responses to self-proteins. When a mature B cell first encounters an antigen that's not associated with an antigen-presenting cell, the necessary co-stimulation is often missing. So if it is not associated with an APC, there's not going to be co-stimulation. Now in this case, the B cell is likely to become anergic or inactivated rather than activated. If it encounters an antigen that is associated with an APC and we get the necessary co-stimulation, that second signal, the B cell becomes activated and is going to proliferate and differentiate, otherwise known as that clonal selection, and is going to clone into plasma cells. This table here summarizes all of the activities of cells involved in adaptive immune responses. So this is a really good table to review if you're not understanding this type of immune response. So we have the cells on the left here, these antigen presenting cells at the top and lymphocytes at the bottom here, and then their functions are all going to be to the right. Really good table to review here. With advancing age, most people become more susceptible to all types of infections and malignancies. Their responses to vaccines is going to be decreased, and they tend to produce more autoantibodies. These are going to be antibodies against their own body's molecules. In addition, the immune system exhibits lowered levels of function. For example, T cells become less responsive to antigens, and fewer T cells respond to infections. Now, this may result from age-related atrophy of the thymus or decreased production of thymic hormones. So the lymphatic and immune system, this has a lot of influence on other systems when it comes to homeostasis. Lymph is going to drain excess interstitial fluid from almost every other system so that you don't have edemas everywhere. Edemas are going to be that accumulation of fluid in other body systems. The lymphatic system also helps to defend every other system against pathogens and microbes. So it is a very, very important system overall. Acquired immunodeficiency syndrome, also known as AIDS, is a condition in which a person experiences this telltale assortment of infections due to the progressive destruction of immune cells by the human immunodeficiency virus, or HIV. So AIDS is the condition, whereas HIV is the actual virus. AIDS really represents the end stage of infection of HIV. Essentially, HIV is the virus that kills all of your immune cells so that you really have a minimal immune system. And then AIDS is going to be the resulting disease. Someone that's infected with HIV, this virus will be present in the blood and a lot of body fluids. So it's most effectively transmitted by actions or practices that involve the exchange of blood or body fluids between people. A person who is overly reactive to a substance that's really tolerated by most other people is usually said to be allergic or hypersensitive. Whenever an allergic reaction takes place, some tissue injury occurs. Now, there are 4 types of hypersensitivity. These are type 1, 2, 3, and 4. They each have different mechanisms of action, and the timing is different. Really not too important for our class, though. In autoimmune disease, also known just as autoimmunity, is when the immune system fails to display self-tolerance and it attacks the person's own tissues. Autoimmune diseases really usually arise in early adulthood and are common, afflicting usually an estimated about 5% of adults in North America and Europe. Interestingly, females suffer autoimmune diseases twice as often as males do. Therapies for various autoimmune diseases will include removal of the thymus gland, injections of beta interferon, immunosuppressive drugs, as well as various other treatments.